in that aim of bridging quantum sciences and biosciences is now not only understanding what quantum coherence might do as a mechanistic phenomena in these systems, but also now with all the advancements of quantum science and technology, how we might be able to use quantum technology, not just to observe in new ways biological systems, but to accelerate discovery in biology, and in particular, to affect biology. And in that respect, one of our directions is, for instance, because we are interested in photoactivated systems, one natural avenue of research for us is what happens if, instead of considering the thermal light that the biological systems that we are interested are illuminated with, we consider quantum states of light, what advantages that might bring to the biology of the system. And a third direction, all of these three directions are not independent, they intersect, but they do require different tools to be addressed, is the biology for the quantum domain. That one we work with synthetic biologists, and the idea is to understand how we can deploy the tools of synthetic biology to manipulate and affect the quantum domain. For today's talk, I'm going to divide it in two parts. I'm going to focus in the first two avenues. First, I'm going to talk about our ideas towards the functional quantum phenomena, identifying, certifying, characterizing without ambiguity functional quantum phenomena in biomolecules. And the second part of my talk will be this aspect of quantum technology for accelerating biological discovery, and in particular for manipulating biology. So let's start with the first part of this talk towards characterizing functional quantum phenomena in biomolecules. And to do this, I'd like to just take a step back and present what I consider is, for me, the core of quantum biology. And for me, the core of quantum biology is that we are trying, the community that has a very variety of disciplines representing physics, chemistry, biology, engineering, etc. What we're trying to do is to do this systematic investigation of the quantum domain of biological systems, and we want to understand its impact, so causal relationships to biological function at different levels of complexity. So I am going to give you an example. In this figure, we are illustrating, this is the light harvesting 2 complex that can be isolated from purple bacteria. It's a protein that bounds around, in this case, the one that I'm illustrating is 27 chromophores organized in a very particular way. In the cell membrane, these complexes follow a particular organization depending on the light conditions that the bacteria was grown. And what you see here is an atomic force microscopy of the membrane, image of the membrane, and it contains the small kind of structures that you can identify there are the LH2s, and it has other slightly larger structures that are other light harvesting complexes that embed reaction centers where the chemistry of the photosynthesis happens. What we would like to understand is what happens, how the quantum processes that happen at this level of this molecule affect the energy transfer across this whole membrane. Of course, we focus first on understanding what happens at the molecular level, but in trying to understand really the impact of quantum biology, that's where we are going. And now, once we understand what is the impact of how that phenomena is amplified, affected, has an impact in this membrane level, then we want to understand what happens now in terms of the survival rate of these bacteria under different conditions. And that can be done in the natural conditions or in laboratory conditions. And that's kind of the thinking we would like to follow in understanding this problem. So this is a multi-scale problem. And we really need to understand that even though we might focus on the molecular level, the goal of this field for it to move forward in a way that will really attract not just more scientists and young scientists, but the investment that is needed to advance this field, is that we don't lose from the sight that this is a core issue in advancing quantum biology. And these multi-scale approaches is relevant, while I'm going to focus here on excitation dynamics, is relevant to the majority of the phenomena that we've discussed here, whether it's excitation dynamics, charge transfer dynamics, vibrational dynamics, or spin dynamics, the one that was presented yesterday, for instance, by Alex and other speakers. And these dynamics are dynamics that are initiated either with light or that are sensitive to magnetic fields, as it was presented. It occurs at the molecular level, but it impacts rates of energy transfer at highest scales of complexity. It impacts rates of chemical production. It impacts rate of gene regulation. And that indeed then impacts other biological functions at a higher scale. So even just by doing the connections of, if we characterize what is quantum of these dynamics and how that quantum might give an advantage to these processes that are usually associated to certain rates, we will see that that is not a simple problem to address. But if we are able to do so, we will have achieved a milestone in quantum biology. So from that perspective, I believe we have two key challenges that we need to address. And my group and other groups around the world are trying to address those challenges. So one of the first key challenges is experimental and theoretical evidence of those quantum, what is not trivially quantum about those processes, those dynamical processes, and in particular in physiological conditions. And now how experimentally we test the impact of this at the molecular levels systematically at these other levels of complexity. I'd like to show you here our attempt. And our attempt is not linear. It's not that we did first this, the second, etc. But it's the work of about 15 years of working in this field. And one of the first things we did long time ago in 2009, just by the time it was around the first QEPs here, was to understand energy transfer in these photosynthetic membranes with a very simplified process that essentially just takes rates that are derived from what the experiment gives you. And then we identify, this is an incoherent process. So its rate is excitation being a molecule de-excited, another one excited, and so on in a random manner, just following thermal processes with rates that you extract from the experiment, but that can be measured, that has performance measurements that can be done in the experiment, like the quantum yield, the transfer time, fluorescence yield, fluorescence lifetime. Now, what we can do now, after 10 years of working in this, is that now we have the whole theoretical framework that allows to identify not just the transfer rate from one molecule, gigantic supramolecular complex, to another one, but internally what is happening with these molecules, which is the first step in the absorption of the photon, and how that is a rate-limiting step for what is going to happen next. So now we will be able to integrate this. I'm not going to show you here results of that, but I'm going to show you how we have achieved now the framework that will allow us to do this multi-scale approach. Now, this is not just of my interest, it's not our individual work. Here I am highlighting the work. This is a paper report, this is our reported last year by the group of Gray and Fleming in California, but they are trying to do the same now for an even bigger system. This is photosynthesis too. So the main message is here. To be able to understand quantum effects in biology, we need this coarse-graining approach and this multi-scale approach. So far, we have focused definitely on the molecular level because there is still a key challenge to solve there. But we cannot lose from the sight that what we are trying to do is to connect what is happening at the molecular level to the biology in different levels of organization. Now, in the rest of this first part, what I would like to focus is to show you that when you consider a little bit more the complexity of the biology of these systems, it starts giving us some insights that are very important for what follows. And I'm going to start with a problem that is relatively simple, but that gives very insightful results. So again, I'm considering here the light harvesting 2, because this is a simulation that a long time one of my PhD students did, and I felt very proud, so I always have to show it. And as I said, so in these systems, you have these 2 rings of very tight-binded chromophores. So one absorbs at B100. This is the one in which the chromophores are more sparse, and the ones that are very tight, where they absorb at a band that is around 150 wave numbers. What you see on the right here is the energy landscape of this system. So it's not a trivial energy landscape, but it has an interesting organization. So it absorbs in states that are in this band, and then it transfers to this band, it relaxes to the lowest energy states, as it is transferring energy, and then before it is emitted, it transfers to another complex, and so on until energy is trapped in a reaction center. Often, what we know for sure that is fundamental quantum and that it definitely affects the function of these systems is the collective nature of these excitation states. These excitation states are excitation states that are delocalized across the chromophores. So they can be partially delocalized, but in general, they involve participation of these states. And we know from experiments, from theory, and it's very well established that depending on that delocalization, of course, it affects the absorption. And in fact, photosynthetic systems have evolved different electronic structures to be able to absorb the full range of the visual spectrum of this system. And it is taking advantage of that collective nature of those exciton states. What we don't know very well is what is happening in terms of the dynamics at that single molecule level. So in order to address that problem, I'll give you some insights of what we are doing here, but I'm going to go back to the biological conditions. We're doing different things. So from the experimental viewpoint, we join efforts with a quantum optics person at the University of Cambridge, and we're depositing individual LH2 molecules on a film, and we're able to do quantum optics on each of these molecules. This has been a long-term project. It's one of the most difficult things to do, to learn how to manipulate this and to do quantum optics for each of these individual molecules. But here I'm showing that we're in that direction. And the importance of doing this is because each LH2 has a slightly different conformation and therefore has a slightly different electronic structure. So differently from the quantum systems that we normally have in a lab, in which they are all ensembles of identical systems, here we have an ensemble of disordered systems. And that disorder in an ensemble experiment then hides that quantum nature that we are trying to understand. Therefore, we need this kind of single molecule experiments to actually understand that quantum phenomena and the heterogeneity of that quantum phenomena. So this is an attempt in that direction. But we're also doing the theoretical aspects of this, which will be what Halman, Talia, and Eddie will explain after my talk. Therefore, I will not tell you, I will not spoil, but the theory in the direction of quantifying this coherent phenomena, witnessing in an unambiguous form, and in a conceptual clear form, is the work that we have tried to do for a very long time. And Halman will present our latest attempt that we are very excited about it. And Talia and Eddie will present our most novel exploration of techniques that are inspiring quantum science to see if we can give other insights to this problem. Cannot spoil the results. Let's go back then to the biological conditions. These experiments are with molecules that are isolated. So essentially what they do is chemically using a detergent. Don't ask me exactly how they do it, but because I'm not the biochemist who does it, but they using detergent, put these molecules in that detergent, and that is able to isolate the LH2, the LH1, and then the majority of the experiments do ensembles of these molecules in that detergent. But one question that all the scientists that have done these experiments and that I have asked myself from the beginning, in the biological conditions, these systems are embedded in a membrane. How different or how good benchmark is when the experiments are done in detergent? Is there anything extra that comes from these molecules being embedded, being precisely membrane-bound proteins? And this is not an easy problem to solve, because when you analyze that directly in the physiological environment that is on the membrane, you then have other complexes interacting, you then have the LH1RC in there. So it's very difficult to isolate exactly what is happening within one single molecule. So there were ideas of what might be happening there and how the membrane environment was important for this system, but it was not possible to isolate it fully, because if you do in these membranes, you have other phenomena, so you could not isolate it. Luckily, this field has a lot of very smart people. And in 2010, Gabriela Schlau-Cohen at MIT, she designed these LH2s that are embedded in a lipid environment that is very similar to the lipid composition of the membrane. The difference is that now you have a sparse LH2s that are not interacting, and therefore you can do analysis at the individual LH2, but also at the ensemble without interactions between these. And this was quite cool because then she asked the same question that people have been asking for a while. Are there any difference when we do this in the membrane physiological environment, or like a physiological environment, or when we do this in detergent? And the main result that she shows is the following. By comparing the absorption in detergent and in the membrane, what she sees is a small shift, of a 3 nanometer when you have the absorption in membrane or in the detergent. And then doing transient absorption experiments, she's able to show that the key difference that is associated to this 3 nanometer shift in the absorption spectra in the band of the BA150 is that you have a much faster transfer rate from the BA100 to the BA150, which is the key step in the absorption of this photon. And it's around a 30% increase in the transfer rate. So in terms of transfer time, she measures that for the lipid environment is around 670 femtoseconds, while in detergent is much slower. And this is important because this internal transfer prevents excitation from being dissipated, but it also enhances the transfer from the LH2 to another LH2, which at the end is the step that matters when it is going to transfer to the reaction center. So this was a very interesting result. And what we decided to do is we joined a team and we said, okay, can we track those changes to a fundamental quantum behavior that is enhancing these transfer rates in the membrane environment? That was the question that we asked. Of course, we have a hypothesis that that should be the case because we know that collective exciton states shift the absorption spectra of these complexes. So based on that hypothesis, I proposed this problem to Chantel. She's not here with us because she's finishing the corrections of the minor corrections of her thesis. She just passed her PhD. And so therefore she's doing that. But we join efforts with her. We join efforts with Harman, who is now a postdoc in my group. And we join efforts with quantum chemists that are at Pisa University, because it's not a simple problem to address. And the answer to this question is yes, we can track this change and this increase in the energy transfer to a fundamental quantum property that is altered or conserved or preserved, enhanced in the membrane environment. And I'm going to show you that here. Because of my time, I cannot just go through all the details, but the methodology is relatively straightforward. Because we have now an expertise in understanding these systems in a non-perturbative matter, because we want to capture the nuances of the details of the dynamics. So we are able to compute the quantum dynamics of the full LH2 system, so the 27 excited states of the LH2. And we compare it with a simplified theory to see to what extent the simplified theory, which is generalized Foster theory, gives us good insight. And when they differ, then we focus as to why they differ. And when they match, then we say, okay, that's great. We were expecting that they match. And we use this theory, and we use the quantum chemical input from our collaborators in PISA, which their strength is to actually generate the Hamiltonians and the spectral densities that are relevant for the problem. And we can track, we can already identify that there is a key change that happens in the membrane structure, depending on the lipid composition, and it's an increase of the electronic interactions between the chromophores that are in the BA100 and the BA150, plus some changes in the exciton delocalization of the system in the BA150. So here is the main result that we have. So one of the main results is that we do the theory at that level of non-perturbative theory, just to make sure that people don't say, okay, this is all to approximate. We don't know. Okay, fine. We do it to a very, as much as possible, accurate detail compared with experiment. And we see that we can predict more or less what Gabriela saw in her experiment. Similar increase, similar shift, but most importantly, because we have now quantum chemical information, we can predict how that shift changes depending on the lipid composition. So we are including now these biological conditions that allows us to see how nature might be tuning that in these physiological conditions. What is the most interesting part is that now, because we have and expertise in quantum science and bringing in quantum information tools that allows us to quantify coherent phenomena in this system. Here we're going to talk about only simply the localization, but it's quantified in a manner that is rigorous enough to tell me if you have more coherence of this kind, it is definitely more of this. In contrast, for instance, if you were to use the inverse participation ratio, which is a typical measure of exit on the localization, it's good to give you a qualitative insight, but it's not a good quantitative measure for what we want to do here. And what we are able to see is we track differences, key differences of what is happening at the microscopic level, the transfer processes that are happening from the states of the B100 to the states of the B150. Because my time is short and I want to show you my time to talk, I'll invite you for you to read this paper. Now, I'm very proud of that paper for two reasons. It was selected as one of the best of biophysical publications last year. Now, this is not because of ego. It is because it is a recognition of the biophysics community that this is giving a new insight into problems they have thought in a different manner. So it's bringing these communities together in at least agreeing on important thinking. And for me, that's important because I think that will move this field forward. So I was very excited that they recognized and invited us to give that talk. The most important feature that I want to mention here is that we're able to track this increase in the energy transfer to a particular figure that is a quantum figure of merit because it corresponds to the geometric average of the average delocalization of the excitons in the B100, the average delocalization of the excitons in the B150. This delocalization is quantified using a state difference, a state distance measure. So that is important because it really more of this implies more delocalization, weighted by the electronic coupling between the B100 and the states in the B150. So it's kind of very intuitive figure of merit here, and it only relates fundamental to the structure. So here I'm not relating to dynamics. It's fundamental to the structure, but it is telling me how the membrane environment has modified that electronic structure to enhance these features and increase that transfer rate. Now, as I mentioned, here we're not dealing with molecules that are identical. So when we do the average, we need to do an average of molecules that are slightly different. So they are accounting for the static disorder. Often people focus on the averages, but we also focus on the distributions of these transfer rates in detergent, which is the blue, and in the lipid composition, depending on the lipid composition. And these are the distributions that are in different colors, yellow and red. Now, as I mentioned, we do comparisons of those theories, the approximate theory and the more accurate theory. And we see that they give us similar qualitative insight. So this is very good, which means that when we want to gain qualitative insight, we don't need to spend a lot of computational resources and time doing these simulations, but we can gain a good insight with the approximate theory. But it's in the details where we will start understanding how other quantum phenomena might play a role. One of the key differences that we see is in the skewness of these distributions, which is telling me that the approximate theory is not capturing properly the rates of transfer in configurations that have these slow energy transfers. But we can see, and this is something that Hallman has done, is that he can track that the configurations that lead to this skewness here are configurations when you have large energy gaps, much larger, between the excitons that you have in the B100 and the B150. So something here is captured by the more accurate theory that is not captured by GFT. Now, the more accurate theory is a theory that accounts for no thermal equilibrium phenomena in this system. And that implies that during these dynamics, it is still able to capture some quantum phenomena. What is interesting is that it seems to be more relevant when we have those large energy gaps. Now, I cannot spoil because Hallman will give you a hint of why this might be the case. So I'm going to summarize this first part of the talk. By considering important physiological conditions, we're able to see that the biology of the system has increased or enhanced quantum features that are fundamentally associated to the structure and that dominate the transfer pathway that increases that energy transfer rate that is measured in the experiment. We have also seen that while we can have a qualitative agreement between an approximate theory description and a more accurate theory description there are still aspects that are captured by this accurate theory that is giving us a hint of where to look if we want to see where there are other phenomena that we might have not considered. And well, I didn't elaborate on this, but the fact that these distributions are changing the width of the distribution depending on the lipid composition and the average transfer time, it reminded us of something that happens very often in biology, and is this kind of trade-offs in between doing something really fast, but then spreading it, or doing something very slow and then making it more precise. And this speed and this trade-off between that speed and the spreading is something that it is observed in very different biological systems. Now, this is just a hypothesis, might not be anything related, but because we can vary the lipid composition of these samples, then it looks like it's something we can investigate. It will require other techniques like laboratory evolution experiments, but it is opening up a new direction of research for that. So with that, I would like to conclude this part one. Before I move to part two, do you have a very pressing question you want me to address before I move to part two? Not many, just one. Just one, because my time is short. So just one or two that you think from this first part before you go to the second part, and I will forget. Do you have one question? Please share. Here behind.
No.
Okay, if you could generalize the findings of this specific experimental setup to phenomena that we can wrap our heads around in the quantum biology sphere, how would you describe it in 1 to 2 sentences?
The one that I have described here?
Yes.
So what I have described here is that the physiological membrane where these proteins are embedded are maintaining specific interactions that when we take them out of that, we decrease those interactions. And those specific interactions are essentially enhancing fundamental quantum features that determine very important transfer rates in these systems. So the experiment is we can do a systematic changing of the lipid composition and see how it changes. And it will be lovely to see how to continue. Here it was for 2, the experiment only did it for 1, it will be great to see it. But Gabriela has done experiments in other complexes. So this applies for LH2, but it's not necessarily something that you can generalize for all the complexes because other complexes have other functions. Thank you. Okay, so I now have 18 minutes, so I'm gonna move to part 2, but do not forget the questions of this part 1, okay? So let's move to part 2. So part 1 was about telling you, like, look, when we look into these physiological conditions, we start seeing things that maybe will lead us to the paths that we will then identify exactly how quantum phenomena is affecting biology in these systems. Now, for this second part, I took a very different approach. I'm interested in systems that are photoactivated, but I also have a quantum optics background, and I'm very familiar with quantum states of light. Doesn't bring any advantages for activating in a biological setting using quantum states of light. Some people will tell me, but why would you ask that question? These systems have evolved to be excited with the thermal light, with the sunlight, so why would they present an advantage? I'm gonna show you that these systems have quantum structures that will benefit from any quantum input that you put on the system. How we exploit that for biology, that's to be seen. So to address this problem, we started with something that is very well known. In the previous example, everything involves the absorption of 1 photon. Now, I'm gonna go to the next level. What happens if these molecules absorb 2 photons? So 2-photon absorption is a well-known nonlinear process that many molecules, atoms, diatomics, organic chromophores, other more complex protein photoreceptors can undergo. And it's a nonlinear process in which you populate a target excited state by absorbing 2 photons. Depending on the structure of your system of interest, you can populate to the first excited state through a virtual state, or you can populate to a higher excited state by populating an intermediate state. In both cases, this is well known, it has been investigated, and it is actually used in biology, mostly to be able to target deep, because it has deep penetration. So if you are using here, normally many of these ones are illuminated in the blue, so around 450 something. But when you do 2 photon absorption, you can illuminate the 800 to 900, so near infrared. That means that you have penetration in the deep tissue. So 2 photon absorption have been used to illuminate molecular systems in biological settings. But it has a problem. To be able to excite with 2 photons, you really need high intensities. And when you need high intensities, in a synthetic system fine, in biology, you essentially are frying the system. So yes, it might have that advantage, but it had the problem of being requiring these very long intensities. Because of that, then people started thinking of, okay, what if we consider entangled photons? This will be photons that are entangled in energy and time, but of course they can be also entangled in polarization or in the special degrees of freedom. Here I'm interested when they are entangled in frequency, because I'm interested in exciting a particular level of these molecules. So the question that we have asked is, will it bring any advantage to photoactivating and eventually generating a rate of a product or a significant rate in biology by activating important molecules with these entangled photons? We don't have yet an answer for that. And what I would like to show you is why that answer might be interesting. So these are results. The majority of the results that I'm showing here are from the group of Ted Goodson in Michigan. These are experimental results. And despite of all the still challenges that the field presents, these are important experimental evidence towards the following. One of the key aspects when the system absorbs 2 photons that are entangled is that it behaves similar to the 1 photon absorption, because the 2 photons that are entangled behave as a single particle. And therefore, it has the advantages of the single photon in terms of being linear with respect to the intensity when it is very, very low intensity, but it has the selection rules that it might inherit from the 2 photon absorption. So it's an interesting hybrid there. So you can already start seeing that other aspects we will be able to explore with these entangled photons. What Ted has shown in this 2022 paper is this is just for imaging. For now, no biology, no activating biology, just to see the biological system. And he is able to show here in his group that when you illuminate with photons that are not entangled, so uncorrelated, and is absorbing at a rate that is non-linear with the intensity, you require a really high intensity, 10 to the 4 photons, to be able to see the system that you are interested. These are cells. I think these are brain tissue cells. But when he takes the same sample and illuminates it with entangled photons, he's able to obtain this image at a much lower intensity. So you are already seeing there is an advantage here. This is just for imaging. Now, when we want to understand what exactly is that the entangled photons are doing, that is very different. And this is one of my favorite papers. This is a paper from 2023. It's the colors of entanglement. And it's a very interesting paper because it compares what happens when we now actually compute the absorption, the 2 photon absorption spectra. So how much is that absorption cross-section depending of the energy of the photon pair? And when you do that with entangled photons, with uncorrelated photons, and you compare it with the 1 photon absorption. And the key finding that they see here is that you see a shift in the maximum of that absorption. So you see that the classical 2 photon absorption has a maximum somewhere in higher energies. And then when you use entangled photons, it is moving towards the 1 photon absorption, but it's not exactly like the 1 photon absorption. And that maximum is shifted towards lower energies. The question is why? And can we deploy this to affect biology? Now, this is a question again that I end up putting very challenging questions to my PhD students, but I'm very lucky because they are incredibly smart. And this is a question that I put to Cristian David Rodriguez Camargo, who just finished his PhD as well, passed, and is now doing a postdoc in the quantum optics lab in Colombia. And so we decided, okay, let's address this question from the theory viewpoint, but let's make sure that we join efforts with an experimentalist who would like to do the experiments that will help us address this problem. And Alex Jones accepted that challenge. Yeah, where is Alex? Good. But before we address that question, there are other challenges in this field that are quite tough and that we need to solve first, so that when we go to the experiment, we actually go with a ground that will tell us, yes, this is what we need to do in the experiment. This is not an exhaustive list of the challenges that are in this field, but it's the list of the challenges that we thought that from the theory perspective, we can approach. So one key challenge in this field is that it is still unclear what exactly from the matter structure will be beneficial in the interaction with entangled light. We know what would be beneficial when we interact with classical light, because two-photon absorption has been studied forever. So we understand very well the selection rules there. We understand that it depends of basically a statistical weighted distribution of the intermediate transition. So that is very well understood what happens. But what exactly from that matter will benefit from interaction with the quantum light? That's not very well understood. Now, this is important for our question of interest, because we would like to know how we affect biology. And the main thing that we know about biology is we can synthesize and we can express particular photoreceptors in systems. So we really will want to say, okay, here is this photoreceptor has this particular structure and it will benefit if it's interacting with quantum light. So that's one of the focus that we would like to contribute to. The second, and is one of the most difficult, is that, as always, there is a lot of different experiments that are not necessarily reproducible. And there is theory, and there is not necessarily a match between the theory and any of these experiments. And the reason for this is because it's not a simple theory to do when you have to take into account the whole complexity of the matter that is involved. And if apart from that, you need to take into account the different experimental conditions, it becomes a problem nearly not easy to address. So we would like to address that challenge. So we like difficult challenges. And eventually we would like to address, to see that possible experimental reproducibility. And that's part of the reason of joining with Alex, that is an NPL, National Physics Laboratory, to see if we can measure, if we can solve also that measurement problem there. Now, in the last 3 minutes that I have before we stop for questions, I'd like to give you a glimpse of what we do. 2 minutes. So the first thing that we do as a theory group is, okay, we're going to propose a theory that can address that challenge. But we need to show you that the theory that we are proposing is well benchmarked and is actually good. So what we did is we consider a problem that someone else has addressed before. And it's a problem in which it considers just a diatomic molecule with a very dense vibrational structure. So it only has 3 electronic levels, but it has many vibrational levels. And the problem that this person addresses, can we target a particular vibrational level with entangled photons? And can we guarantee selectivity of that level? And to do this problem, these scientists address it in a kind of exact form. So for that, you need to solve too many, discretization equations. So the challenge that I propose to Cristian David is, I'll give you a prize if you can come out with a perturbation theory that is more efficient, but it reproduces exactly the same. And because he also likes challenges, he did it. And then we proposed that theory that is reported in this paper. And here we have the results that recover exactly the same that was published before, but with a theory that was more efficient and that allows us to gain additional physical insight that it won't be possible if we didn't have that efficient theoretical framework. And the insight that we gain here, and with that I am about to finish, is that we now are able to see that, yes, if you have a large degree of entanglement, you can select almost any vibrational level that you want in any form. And that opens opportunities for biology because it means specificity. But you not always will have perfect entanglement and the highest entanglement possible. So what happens if that entanglement deteriorates? And what we see here is if you have now entangled photons, but not to entangle, like this is what is showing here, then we see that you select better the low energy vibrational levels. And that is key because it explains that shift towards low energy that the experimentalists in Michigan observed. So we were very excited with this. We said, okay, will explain that. We considered the system that the experimentalists considered in Michigan, and we actually showed that indeed, when you have a large degree of entanglement, you observe a shift that is very similar to what is in the experiment. Now, because these are just results that are about to be reported for publication, I will leave it there with a cliffhanger. Look for the paper in the archive, not too soon, not too far in the future. So the summary of this part 2 is we have developed a theory that is predictive and that will allow us to account for experimental conditions, but also the matter structure, and then understand what aspects will be beneficial to take advantage of that quantum light matter interaction eventually for biology. So with that, I would like to finish, not without inviting you to 2 things. Alex already talked about this. So the GRC in quantum biology, which we hope to see you all of there, it will be in March next year from the 7th to the 12th. I am chairing that together with Alex. But we also have the GRS. And for all the early career researchers that are here, it's a very, very warm invitation for you to attend. The GRS is only 1 day. It's targeted for you. And it's about for you presenting your results to your peers, but also have mentoring opportunities and kind of start building. Me, I'm towards my, the end of my career, not too soon, but you know what I mean. But the early career researchers that are here and the community are the ones to come to advance this field even better. So please join us. Thank you very much. Thank you to the Gordon and Betty Moore Foundation and APS Access.